All right, good morning. We have a last item here as far as the lab practical is concerned. This is a voiceover of the document that I had posted for everybody. And this is my attempt to try to do, I had asked folks to sign up for discussions, and I know that everyone's busy. And so I figured the best way to do this is a voiceover, and so you can watch this at your preferred timing. So what we're going to cover for this lab practical is unit 7, 8, 9, 10, 11, and 12. And you can see the corresponding lab manual pages. And I have them all here. So I'm going to kind of just briefly go through these and point out items that would be good to review for the lab practical. The learning objectives in general are a good thing to kind of look at to see if you're at least aware and knowledgeable about aspects of these, such as know the steps and phases of interphase, and we're going to go over some aspects of this. Occurs in each step of the cell cycle, and then we'll talk about differences between mitosis and meiosis, and talk about karyotypes and DNA replication and that sort of thing. And the manual does a really good job of just reviewing the steps and stages. The prophase, metaphase, anaphase, and telophase, if you recall, are the main steps of the mitosis. And it would be a good thing to remember somatic cells are body cells. The sex cells are, of course, the ones that are haploid. And if you could associate half, that's half the complement. So normally we have 23 chromosomes with a total, because there's two each, of 46. And haploid means that our full complement in the sex cell are 23 chromosomes. In somatic cells, we have the 46 chromosomes. And I just wanted to point that out. I think that's obvious. And most likely you'll see something like that on the lab practical. And meiosis, of course, is in the sex organs, the gonads, or however you want to put it. And the outcome of mitosis are two cells. And just to identify it, the end product of meiosis are 4 haploid cells. These are diploid, having both. So this is 46 chromosomes, this is 23. And so that's the big difference. So if you want to look at a process and what it's ending in, if you look at 4 versus 2, then you'll know. A zygote is the creation of first cell. A new offspring is called a zygote, so that's the new cell. And I think we've been over this enough. Interphase, prophase, metaphase, anaphase, telophase, cytokinesis, and just kind of going through. The cell cycle is really straightforward. We have the G1 phase. This is normal operation. The S phase is the DNA replication phase, and so at some point the timing is such that the cell decides that it needs to replicate, and this timing is different depending on the type of cells that we're talking about. Hair on your head, producing hairs, versus a hair on your eyebrows. If they were at the same rate, it would be trimming your eyebrows every time you get your hair cut, and that wouldn't be good. So the timing is sort of important. All of this, of course, is called the interphase, so the G1, the S phase. And then when we finally end the synthesis phase, and that's what the S stands for, synthesis phase, and then we're just replicating the DNA. So now G2 phase is the part I always hate in a move, is getting everything ready, you know, the packing and all that stuff. And that's really what's going on in the G2. It's preparation for the mitosis, which is the prophase, metaphase, anaphase, telophase, and then you go into the cell division. And so it's really straightforward. So this is the packing stage, and the chromosome, actually you can start to see a little bit more. So the interphase, prophase, you can actually see the DNA, so it's like the boxes that you packed. And then you go into metaphase, where they line up in the middle. So if you can remember metaphase and middle, that would be great, and that's really easy to pick out in any slide. The anaphases, if you remember, I had talked about anaphase, is you can actually make the letter A as the DNA is being pulled and across from each. So anaphase, and you can see that. And we did that in root tip, and then you can do this in whitefish blastula. We had done that in the lab. Metaphase in the middle, just like we talked about before. Telophase, just like in these cells, the telophase, we start to separate, and we start to get a pinching in of the chromosome of the cells, and you can see that plate forming here, and that's where telekinesis. This is in plants, of course. It says here it's root tip, and that's of course a plant, and this is an animal cell, the whitefish blastula, and you start to see the indentation here, and that's the telophase, early telophase, and then late, as you can see, it's clearly two. So we have the metaphase, the anaphase. This is like really early anaphase. This is about the best picture I could find. The anaphase, this one is sort of like a late anaphase, early telophase, and you could make those little A's like I was talking about in the previous. But the interphase, and the difference that with prophase, you can see the boxes for the move. The chromosomes are actually apparent, and here you really can't see much. So prophase, metaphase along the middle. This is really, to me, more of a metaphase, but it is starting to be pulled, and you can see an early telophase. So if you were to label this one anaphase, I would actually think that's better than early telophase. This is sort of like late metaphase. Just to be technical about it, I disagree with whoever put this together. That's okay. It really illustrates the point. Anaphase and then the telophase, you can see they're separated, and we're starting to see the division here. So it's sort of like a late anaphase, and you're starting to get into the telophase, like they were saying, and then late telophase. So that's pretty much it. It's really straightforward, and the images that you'll see in the lab practice will be really straightforward, probably more straightforward than that one. Also, we talked about karyotyping just really quick. It's just where you break out the chromosomes, because what you want to do is prepare. You get the blood, and then you go through various stages of treatment with chemicals to kind of lock them into place. And I've already discussed the haploid and diploid and all of those sorts of things, but the karyotype is actually looking at the chromosomes after using the staining technique and isolating those, and the karyotypist now puts these in to the right type of chromosome, chromosome 1, 2. It's very tedious. If you remember in the lab, we did a really tedious time with that, and the karyotyping. And so this would be normal female, because you have two X's. And if we had one X and one Y, the Y is usually just a small chromosome. And if you saw that, then that would be just the one Y and one X, and that would be male. So this would be the Y, and the X chromosomes are there. And so that's pretty straightforward. And here's a male. You can see that one. Then please review the review questions During which stage of the cell cycle do chromosomes replicate? And I think, if I can find the diagram here really quick, of course, the S phase, DNA replication occurs at that phase. How does cytokinesis differ from plants and animals? The plants, you have that thicker cell wall, so you get more of the this plate that forms in between, where in the animal we don't see that, and it's more of an indentation and then they pull apart, and that's sort of the difference there. And it's really due to the thicker cell wall of the plant, where the animal cell doesn't, there's no cell wall like a plant. It's a thin membrane, and it's kind of the biggest difference. In mitosis, begins with the parent cell. The resulting daughter cells are exact copies. What's the purpose of mitosis? And that is to replicate cells. Full complement of, I guess, somatic cells, body cells, not sex cells. The centromere is located in the center of the chromosomes. What stage of cell cycle spend the most of their time? Interphase. This is the difference between haploid and diploid. This is 23 chromosomes total, and this is 46, and that means that there's duplicates of the 23 that makes the diploid duplicate, if you want to look at it that way, and this is half complement. So chromosomes, or 23 pair. Don't let that get you confused. It's 23 pairs. This is single chromosomes, and so you can see that. What's the purpose of meiosis? To create sex cells with haploid condition of the chromosome. So it has to go through a reductional division to do that. When during meiosis, it's at the end of the stage, is the final additional reduction of the number of chromosomes. So we just go through an additional division, and so there's a reduction division of the chromosomes, and that's how we go from the 23 pairs, how we end up coming from the 46 altogether. So 2n is 46, so the n, the chromosomes, there will be, how many chromosomes will be in a daughter cell? It would be 23 pair, or 46 total chromosomes. And in gamete, which means sex cell, is 23 chromosomes. That's half the number. So why must gametes only have half the chromosomes? Because in a genetics experiment, you have the male and female experience creating a new child. And that means 23, chromosome 23, creating now a new diploid organism with 46, and that's all that's referring. Why does crossing over occur? Is because you have single chromosomes, and unlike mitosis and meiosis, they're able to form a chiasma or to cross over, which just means exchanging legs of chromosomes, which means that's the sister chromatids of others. if you did it on the same chromosome, they would be identical and there would be no blending occurring. Genes and DNA arranged in chromosomes, what can go wrong with crossing over? You can get partial or duplication, all sorts of other types of aberrations. Prokaryotes are able to reproduce as little as 20 minutes. Explain process of cell division be quicker than in eukaryotes. In bacteria, you have a single chromosome, and let's just draw a bacteria cell, and they attach, the chromosome attaches to the membrane, and it duplicates but at a different attachment point of the cell, and then they divide right between those two to create two bacteria cells that have their circular chromosome, so that duplicating, so it divides right through here to create separates the two cells, and we get the two chromosomes, and it can does it sometimes as quickly as 20 minutes. There are other organisms that's slower, but that's okay. Karyotype is just the ability now to stain and to analyze the chromosomes. Full, these would be the diploid chromosome set. Y chromosomes studied during metaphase, they're easy to obtain at metaphase. They'll have the complements. In karyotype, 3 characteristics used to identify homolog chromosomes is size, position of the centromere, and what else could there be? There's banding patterns within the chromosome. Is the way it stains, and it's always the same for each unique chromosome. Which karyotype sample if you remember in the lab you did one. You don't have to worry about answering that one. So the idea is you want to be able to identify if there's a normal is XY male, XX female. If you have a trisomic 21, so you have 3 copies of the chromosome at position 21 or the 21st chromosome. That's Downs as an example. Down syndrome. And that's all I'm going to ask you for on that. Microorganisms, unit 8. I'm going in order of the unit number, not the way we did them, which is fine. I have some videos at the very beginning of this section on microorganisms, and it really will help. And I posted it either a PDF or Word, and you can just copy and paste these and just review those, and it really helped through all the different definitions and things like that. So autotrophs synthesize organic molecules from sunlight, photosynthesis, or they can do it from inorganic chemicals. We call that chemosynthetic. Heterotrophs are acquiring energy from living organisms or decaying, saprophytics or parasites, and you can review that. But a lot of the associated microorganisms can be associated with really awful diseases like cholera and anthrax and MRSA, methicillin-resistant Staph aureus, salmonella, and things like that. And just a general discussion of where they can benefit. And a probiotic is a beneficial where they can grow in our gut. And bacteria actually provide us vitamin K, vitamin B, and various other things that grow. And they also help defensively. They're sort of part of our immune system, where they take up a space where a bad microorganism might take. So the probiotics are sort of like blockers, and they help keep the gut balanced. And if you are taking an antibiotic for some sort of disease or some infection or something, it's probably a good idea to eat some yogurt or some way to replace some of those probiotics, and you'll get back on your feet faster. The microorganisms are used obviously in the food industry, the production of vinegar, pickles, beer, wine, and that sort of stuff, so you can look at that. Prokaryotes, just general features of bacteria, and just read through that. The basic shapes are the cocci, the round diplococcus or strep is in a chain. Staph is in a grape-like cluster or grouping. Then you have Sarcina, which is sort of a tetrad organization stuck to each other. Bacillus are rods, and you can see they can make chains or single, or they can be spore formers. A typical spore former might look like this, and it's got a really dark center, so it's a spore, and that means that they're resistant to high temperatures. So when you see those movies of folks going into Egypt and getting the curse of the mummy, no, what they did is they took nasty bandages and they unwrapped them, and they got in the air spores from Clostridium botulinum or some nasty organism, and they got sick from it. And what do you expect? There's no curse other than the fact that they're filthy, and spores can last a long time, and as we've seen that. Vibrios are sort of like comma shaped. We have sporilla and spirochetes like Treponema pallidum that causes syphilis, or Borrelia burgdorferi that causes Lyme disease, those sorts of things. Helicobacter pylori is a good example of a sporillum that causes ulcers, and typically get rid of it by just taking some tetracycline. So we can also, to treat these infections and the like, we can use antibiotics, antibiotic sensitivity tests, and looking at the zones of inhibition, using antibiotics for that. And the gram stain is a very easy and straightforward test. When it was developed, had no clue what it was doing. It was just finding out that the result were pink or some were purple. The purple are positives and the pinks are negatives. I usually just refer to them as light red because if you can remember, purple is positive and then the light reds are negatives. That would be helpful. So there's a diagram for that. Here's sort of an outline of the gram stain. We apply crystal violet that binds into the peptidoglycans, like a really thick layer that the gram negatives don't have. And then the iodine acts as a mordant that leaves that stain in. And then we destain it with an alcohol. If this can't get dislodged, then that's a gram positive. If it does get dislodged or discolored, then we come back with a counter stain called saffron, and that stains it pink or that light red. And so that's, you see it. But it also stains the positives of that light red, but you don't see it because the purple is so dark, it masks it. So the idea is the purple are positive, and the negatives that get destained, those are the gram negatives that we're talking about. So you can look through that. The antibiotic sensitivity testing, as I mentioned before, the zone of inhibition is where the bacteria are resistant here to, with a lot of bacteria growing on a plate, they'll grow right up to the disc, where if it's sensitive, you'll see the zone of inhibition around it. So these would be perhaps a candidate to give a patient because the bacteria can't grow near. What's not obvious in this is just a piece of Whatman blotting paper cut maybe with a hole cutter, a paper hole cutter, just to make that little insert with Whatman blotting paper. Then they soak it in a particular antibiotic. It diffuses outward. So here's the disc. It'll diffuse that antibiotic, so the concentration of the antibiotic starts to go down as it leaves the disc, so that big circle. So it's starting to get lower and lower concentration. That's why you get this zone of inhibition, and that's useful to determine which antibiotics that we may or may not want to use. So if you were to look at this, and there are some criteria that we use, but we're not going to worry about that here, but A might be a really good candidate for a patient, because you see the bacteria is really sensitive to it, versus maybe E or F, where the bacteria is totally resistant. You wouldn't want to give these antibiotics, E and F, bacitracin, and erythromycin. It's going to be useless, totally useless. Where A, B, C, and D might be useful, the neomycin, gentamicin, vancomycin, ampicillin, to treat this patient to get rid of that infection. It could be a urinary tract infection, whatever. And this is how they determine that. And then you can read through these types of questions and find that in there, in the notes. So these are pretty straightforward. Disease transmission, well, I don't think I need to go into that too much, because we're always kind of worried about that now, about wearing masks and gloves and the like to try to prevent this transmission. But when we sneeze, we create, of course, an area of vomite that can be transferred in others. That's why you always want to kind of sneeze into your elbow, and that way you get it on your shirt and then you wash your clothes. But a face mask, if in the event of like this viruses are different from the bacteria, of course, and but the idea is you want to limit physical contacts and things like that, and they give the influenza virus as an example. But bacteria kind of do the same sort of thing, but not as perhaps as easy as viruses, because their size is so small. I always try to do fist bumps, and or just acknowledge by kind of just saying, hey, you know, how you doing, and not doing the handshake thing, especially in these days. We just don't want to do that, and it's kind of hard. You learn to do that, and it's kind of hard to break those habits, but it's a good thing. But the idea of epidemics, or the folks that do the public health, somebody is spreading the microorganism around, and you can have to actually follow, if you remember or read about CDC, following who the original the person, or the person zero that started to, or that had the infection and now spread it to all these other people. And this is what we're trying to eliminate now with the passage, and that's why we're at home and doing all the things that we're doing. So look through the comments on the transmission there. The other one that we worked on was the crime scene analysis. We had broken that up into two parts, and the polymerase chain reaction was the key part of the crime scene analysis. And just as an aside, they're using PCR to identify the coronavirus, and so they use the same kind of technique. There are some huge differences. I'll just make a really quick reference to as far as the coronavirus, but it relates to exactly what we were doing. The coronavirus is an RNA virus, and corona, the one that we're worried about, and I'm not even going to worry about the name. It's an RNA virus, and it's got to go through a process in order to get PCR to type to work. We use reverse transcriptase to create now DNA. So an RNA virus, and this is why RNA viruses are more dangerous, because that RNA can mutate so easily. It's a single copy, if you recall from class. It's a single strand of genetic material, and it doesn't have a copy like DNA does. So if you damage something, you can kind of look at its partner and find out what needs to go back. Well, an RNA don't, and they tend to mutate a lot more, and that means that it makes it hard to make vaccines if the virus is mutating all the time. But in order to identify them, reverse transcriptase gets it in the DNA, and then we can now use standard PCR just like we studied. So I just wanted to bring that up so you can apply the knowledge that you already have. It's just a little twist here with that reverse transcriptase. So the PCR, the basic steps, and I think I included a flow chart for that. It is in your manual. Let's see if I have that with me. I may or may not. It doesn't matter, because we've covered it so many times in class. But please watch the video, and you can kind of go over. But the basic steps of PCR uses temperature. So we raise temperature, slow it, bring it down, and then we raise the temperature again. The idea behind that is you separate the DNA double-stranded DNA into single-stranded DNA. Then the primers. The primers are the key part of PCR, because the primers are single-stranded DNA, and they, if you have a gene, let's say, that we're interested in DNA, and we're interested in just this region of the DNA, so you have to have a primer that goes upstream and downstream from that gene, so it sort of encapsulates that. So the primers you want kind of to go on either side of that region of DNA that you want to amplify. Primers, and the primers are the single-stranded, so they bind the single-stranded DNA. So we heat to separate the strands, we cool just a little bit, and that allows the primers to anneal, and then you start the heat process again for the to fill in using an enzyme that fills it in, and it has to have that 3' hydroxyl group we talked about, and the DNA polymerase. But it can't be any DNA polymerase. It has to be one that was isolated from an organism that actually is able to grow in really hot environments like hot springs, and in doing so it evolved a DNA polymerase that can handle this higher heat, and in doing that we can now replicate and fill in and now make 2 strands of just the region that we want, and just make lots of copies. So that's the primary purpose of PCR, is to amplify DNA of our choice based on the primers. The primers are the key piece of the DNA, as in doing like the RNA virus, getting the DNA from the reverse transcriptase. We have to get the primers that are up and downstream from that particular pathogen that we know about, so we can amplify the DNA. If you can amplify the DNA, it means that the virus was there. If you can't amplify it, then it's a negative test. Anyhow, that's the key with that. So that's PCR. Then we ran the gels to look at the DNA as we amplified using the PCR. So electrophoresis uses a charge, and we always run to red, which is the positive pole. So you want to load your DNA at the negative end, and that's usually the black pole end, and they just use the red for the positive, black for the negative. And so it'll migrate. So the DNA will actually be pulled due to charge, because DNA has a slight negative charge. It's attracted to the positive, so it's being pulled. And a gel actually is sort of like a matrix of agarose, which is seaweed, ground up seaweed. You heat it and cool it, and it makes a sort of a congeal, and so larger pieces of DNA will try to matriculate through that matrix. And the problem is, the larger pieces is not really a problem, but if you were to run a gel, larger pieces tend to migrate slower, and smaller pieces of DNA will migrate faster. So on a gel, these are smaller and these are larger, and you'll see on a gel, and you separate DNA based on the size, and it's also proportional to charge. Of course, the larger piece will have more charge, but what really the factor is going through that matrix of the gel, and so the larger pieces slowly migrate, faster ones go through it a little bit faster, and there you go. And so you can look at that. And what we do is we amplify a particular region for the crime lab, and so the TH01 locus is one that the police use of many. I guess they have like 12 or 13 of them now, and it's a repeat. It's TCAT, and we all have our unique number of these repeats, and so one from your mother and one from your father. So what we're going to see, if we do it right, are 2 bands representing one from your mother and one from your father at different lengths. and that's like a fingerprint, and it's unique to each of us. And so we use the PCR to find that particular series of repeats, and we go upstream and downstream from that, and we just kind of clamp that region that has that number of repeats, so like in this case, and it'll just amplify that region, and we can now just determine how long it is on size, is that's what DNA gels will do. And so when we run the gel, we'll see two bands, and one usually is larger and one smaller, and it's unique, and so you can size those, and that's what we're doing with the crime lab. And so crime labs, it doesn't take much. It could be a little bit of spittle in a drink bottle. You can run PCR on it, amplify that DNA, and find out it'll have that fingerprint, that pattern, TH01 or whichever one you're using, and it's really easy to do. Even leaving fingerprints on windows and things like that could actually generate some DNA. Hair from a hairbrush, things like that. And then we run the agarose gel. We also talked about micropipettes, and you got to make sure the tips are on those, and then you can set the micropipette to a particular volume by rotating the adjustment thumb screw here. And on the front of the diagram is that you have of your pipetter, you have various different ones. We use the P20, the P200 and the P20, and so the P200 had the three solid black numbers, and this would be read as 125 microliters, where the P20, the last character here, will be in red, and so this would be 12.5, so that for the P20. So we can pipette, and the ranges you can see the recommended. Now we only used the P20 and the P200, so there's the 220, and it shows you what it can do, and you can read through it, but everyone's already done that technique, so you should be aware. There's a good diagram of the PCR. I guess I stuck it in the wrong place. It doesn't matter. So you can go back and review from that chart in that section there, but you take the double-stranded DNA, you heat it to separate it, and then allows you to adhere the primers. Now those are the primers. And for TH01 for the crime scene, we know what those primers are, so we're only going to amplify that region of repeats, and we'll determine the sizes of those. And you can see more of a breakdown of the diagram of that particular process. And then the electrophoresis that I've already discussed with you. And it's just a diagram. The DNA will be pushed through. We always go from the negative electrode to the positive. You run to red, and then the DNA will separate out. So in the crime scene analysis that we did, usually we run a ladder to estimate the size of the DNA, and it's not always necessary, but it's good form to do that. And there's the crime scene. So there's DNA that was found at the crime scene, and then what you do is you find of the potential suspects, do any of these suspects have DNA that has those fragment sizes that are unique, like a fingerprint, one from the mother, one from the father, that line up with those that were left at the crime scene. And sure enough, B does. And we had in this gel we have the crime scene, and we see suspect C seems to match on this case. So you get the gist of that, and you may be asked to do this on a lab practical, which is really straightforward, and just find those. I don't think I need to go through this. Everything that you need is in the handout, or this manual to answer these, but it's important to review those. That was an extra page there. So we're going to go in the crime scene analysis part that we just discussed. You've got all that. And that's a duplication there, so we don't have to worry about that stuff. And we covered the microorganisms for that section, and we're in good shape. So the evolution and those parts that we covered, those were the newest ones. Evolution and ecology were really straightforward, and you can review the handouts that I provided for those, and the voiceovers I've already done for each of those sections. So I would recommend, instead of generating more video for you to rewatch, just pull out the voiceovers that I have already done for the ecology and evolution and the genetics, and for the reviews on that. And the portions of the lab manual are provided, and you can find those on Blackboard within the laboratory area or the laboratory materials, and you can see the various units and the names, and just review those voiceovers for those and as a review. So that should get you covered for the lab practical. I posted some questions that have answers, or sample type of lab practical questions that you might see. You've already been through a lab practical, so you kind of know what to expect. But on an online, I'm going to make it as straightforward as possible. I'm going to post the exam the beginning of next week, and I'll send out a link to everybody, and that link will take you to the exam, and you'll have I think it'll be 50 questions, and you'll have an hour to complete it, and I'll allow you to go back over it and make sure that you've answered everything the way you want to, so you can change your answers if you want during that hour. And then when you're finished, you just click finish, and that'll be it, and it submits it, and I will post the grades as fast as I can. But I think this is the most concise part that I can do for the lab practical. You'll have all the sections that are posted, and for redundancy's sake, I just leave it to those. So just be sure you can review through those sections there. If you have questions, just feel free to email me, and I'll do the best I can on getting back to you timely and to answer those. So thank you. I hope everyone's staying well and through this period of time, and I hope this was useful for you. And I'll be talking about exam 3 for those who are going to be taking exam 3 with me, and then the final exam as we move forward. So we're getting down to the end here, so you're doing great. Keep it going. I know you might feel overwhelmed for some, and just push through. I'll help you as best I can, and we'll go from there. Thank you.